Good afternoon. I am Dr. Gualberto Ruaño. I'm at the University of Connecticut at the Connecticut Convergence Institute for Translation in Regenerative Engineering. It is my pleasure and my honor to be your last speaker for this series of 4 webinars. The slides show some of my biographical details. The title today is going to be Personalized and Precision Medicine Approaches in Regenerative Clinical Trials. This talk will be over a period of about 1 hour. We'll break at the top of the hour for questions. Please use the chat tool to send me comments or questions as we go along. The chat, this conversation, this meeting is being recorded, so you should be able to avail yourself to it later at the Connecticut Convergence Institute website. This has been a partnership with ARMI, Advanced Regenerative Manufacturing Institute. Yukon Health is a Tier 1 member of ARMI, and this activity is part of our cost share, where we bring to the community resources, knowledge, expertise that will enhance the entire industry and the entire membership of ARMI. So it has been a great ride this month of November, and I'm privileged to close the series today with this topic. So in terms of a little bit of background on myself, I have been in the field of personalized medicine now for my entire professional career, which is now spanning 25 years and counting. And I have worked in a number of areas within personalized medicine, and that includes diagnostic applications, therapeutic, policy, and also, very importantly now, of course, academia and teaching here at Yukon. So I train at Yale University, where I was a fellow of the medical scientist training program. And I have been now different phases of my career in the pharmaceutical sector, where I began after I finished my studies at Yale, Genesys Pharmaceuticals in New Haven, which was one of the first, if not the first, personalized medicine company. Then at Hartford Hospital, where I had a clinical laboratory called Genomas that worked very closely with psychiatry and cardiology at Hartford Hospital. And then over the last 4 years here at Yukon, working with the Connecticut Convergence Institute. So the topic today is one that I will address by almost presenting a new dimension to the thinking of the organization. And that is that the variety of responses that we see in health to any intervention can be, first of all, recognized, and secondly, it can be utilized to drive learning and drive precision in the management of medical conditions. In our field, in regeneration, we know about the lineages, we know about the possibilities of having stem cells differentiate into different cell kinds, and we have precision in the sense that we can profile specific markers that would allow the cells, in this case mesenchymal stem cells as an example, to differentiate into osteoblasts, adipocytes, chondrocytes, myocytes, fibroblasts, or also preserve the function of self-renewal so that the cells can perpetuate themselves. And we have means of profiling this. The dimension that I am adding to this is for us to start thinking of the variation in those mesenchymal stem cells or those progenitor cells. And this is recognized by the fact that the genetic background of the donor of each of these cells must be taken into consideration as we evaluate the destiny of these cells, whether they renew or whether they differentiate there is a background genome that is inherited by the donors of those cells. In addition, there are epigenetic modifications in that background that could be occurring in the cell culture conditions or in the harvesting of the material or the implantation into a recipient. So rather than think that, stem cells are all the same, we have to start considering the fact that they are not, and that the donor, the variability in the donor carries over to the culture. There is a recent book which is one of the first I see to tackle this issue of heterogeneity, and it is shown here. It's called Stem Cells, Heterogeneity, Novel Concepts. It's a series of 3, which I found to be very informative, and I will refer you to this compilation of articles in those books for additional information on this topic. So I want to begin by presenting a sort of very personal view of how variability and genetic variability can lead to precision medicine. And it begins with an evolutionary perspective. When I trained at Yale, my background was in population genetics and evolution. And this is something that continues in my thinking as I think about the reality of modern healthcare in modern populations. And that is the genome that we carry is really a legacy of previous generations that have led to our current species. And each of those variations can carry a sort of adaptability to the environment. In some cases, the environmental variation can have different effects on the ancestral conditions versus the modern conditions. For example, if you look at drug metabolism and the cytochrome P450s, those are among the most variable genes in the genomes, similar to HLA in terms of having multiple forms or multiple variants. And that is because in ancestral time it had advantages with regards to toxins, to process plant and environmental toxins. In the modern world, we see implications of those polymorphisms in the metabolism of medications, because as far as the body is concerned, these medications are foreign substances that have to be basically detoxified. And that is what we see now with pharmacogenetics, where a lot of the variation in the response to medications can be traced to these cytochrome P450. That's just an example. On the metabolic pathways, you can also see variants that may have been advantageous in the extraction and retention of energy. And those same variants in the current world may have basically negative implications if they induce obesity or metabolic disorders, where you may be basically using the same ancestral genes, in this case, to maintain energy that is not required and needed, and then it evidences itself as obesity. So all I'm suggesting to you is that many of the polymorphisms that we have now have reasons to exist, and those reasons to exist will tackle or will tip the response to different interventions one way or the other, depending on the impact of the stimulus that we submit to those organisms. We will then see the effects of these modifications or variations that we wouldn't otherwise see. And so in the context of definition of precision medicine in terms of operations, I think that a lot of our thinking in terms of interventions in health is related to the average responses to a treatment. And we try our best to define that variability around that average using standard deviations and other statistical tools. But we always, most of our thinking has always been based on this average. And what I have been advocating is that we start to think about precision medicine in the context of personal health. And that is, rather than say, look, you belong to this population, therefore we will predict that you have this average response, that we will be able to say you are in a different sector of the population, where in the case of, let's say, a risk to a medication, you may actually be on the higher end or the upper quartile of the risk of side effects in this case, in this example. So please observe the differences in thinking from the left, where we say you are average because you belong to this population, to the right, where we say you are actually on the upper 25% of the risk based on your profile, genetic profile, phenotype profile, etc. This is what I believe is the fundamental concept that is driving precision medicine, has a predictive model for responses in healthcare and medicine. So the way I have approached this is I published this 15 years ago in the Biomedical Engineering Handbook, and it was reviewed and revised in the 2015 edition. And the basic model or the basic paradigm is, okay, so let's begin with variation. That is the way it is in the context of responses. Can we learn from that variation? And that is shown in this curve. So imagine that you are on the left side of this curve, and then you profile the variability in the frequency of a polymorphism as you go to the upper side of the distribution. So most markers, think about genetic markers, think about polymorphism. If you look at the frequency of a given marker, as you go from the low end of that curve to the high end of that curve, most markers will be the same in terms of frequency. The gene is there. What I'm asking to do in this thought experiment, and I'll show you actual examples, is we can scroll from the left to the right and calculate the frequency of that polymorphism. And for most markers, it will be the same. Doesn't matter if you're on the left side of the curve, the center of the curve, or the right side of the curve, the average will be the same. The frequency will be the same. Some markers will have a very different profile, and they will have a low response, a low frequency on the low end of the curve, and a higher frequency on the right side of the curve. Why is that? Because they are tracking with a higher position in the distribution. And that is what we would call an associated marker. Conversely, there will be some other markers where the frequency actually decreases. The frequency of the polymorphism decreases, or the intensity of the marker decreases as you go from the low end of the distribution to the high end of the distribution. This one is also happens to be negative as the outcome. This one happens to be positive with regard to the outcome. And this is the way that we actually can learn from variability. So the way I have used this paradigm begins with the use of very specific genes that have been selected based on their function. The work I have done, it has been in the area of cardiovascular and mental health. So we look at genes in these pathways. So these are so-called candidate genes, and these are basically related to mechanistic insight. They have been selected based on their potential role in a given pathway or a given phenomenon. And you could do, in the series of research that I will be describing, we use 222 genes derived from these pathways. And then we apply this to many different problems in healthcare: drug response, imaging, diet responses, exercise responses, surgeries related to bariatric surgery and weight loss, even populations, looking at admixture in different populations in Puerto Rico, for example, that have admixture and presence of multiple traits from multiple origins. So it's quite a range of interventions from drugs to imaging of the brain, and all are using the same concept, learning from variation in response. I will focus as an example on the exercise, because exercise, we know, is also regenerative. And in fact, it's a great means of prevention of disease because of the way it conditions the human physiology, and in some real senses, it actually is regenerative. So it begins with a very detailed profiling. A study that was a relatively small number of volunteers, but this number of 100, 200, 300, that is the sweet spot for most of the clinical trials in regenerative medicine. I think we discussed this at the last seminar on statistics. So this number 100, 200, 300 is the number to deal with when you're dealing with discovery based on different interventions. So in this case, the intervention was exercise, and there was 40-minute bouts 4 times a week. And then we did genetic consent, and then we measured a number of cardiovascular phenotypes. And then we look at the heterogeneity in responses. This was published in 2004. This was done with Dr. Paul Thompson. And here are some of the measurements that we do. Very, very extensive profiling of lipids, plasma, lipoproteins, and then the phenotypes at the organismal level or the body level in terms of weight, body mass index, etc. So this shows you that you can do very deep profiling at the phenotype level. Before you do any genetic analysis, the profiling of these biochemical and physiological markers is key, and this is something that we can adapt in regenerative biology as well. So look at BMI and HDL, and you are seeing this distribution. In my assessment, anytime you see variability, anytime you see a distribution, that means you are in business. It means that there is information that can be gathered because the response to the same intervention was very different. So you begin with the phenotype, the baseline BMI, you see the average was about 27. The change in the BMI after the exercise intervention, you see that for most people, there was a reduction in BMI. The mean is 1.19 negative. However, not everybody responded the same way in terms of BMI. Some folks actually gained a little bit. Looking at HDL, which is the so-called, quote, good cholesterol, unquote, the average was 48. And then the actual mean on the response was an increase in HDL, which is what we know exercise can do, an increase of 2.28 in this cohort of 119 or 119 individuals. But again, some people did very well. Some others, the HDL actually decreased. So this just is to illustrate that even an intervention as well regarded as exercise is going to have positive effects on most people, but not all people, and that the variability is quite substantial, even in a very controlled environment, as was done in this protocol. So the question is, okay, so that is the way it is in terms of variability. Is there anything we can learn from it? So I'll take you through a genetic analysis of this, and you will see how some of the genes actually correlate with the response. So again, the analysis was on a set of 222 genes, and here are the ones that showed the responses I was mentioning to you, the ones that have decreases in frequency as you go to higher responses, or the ones that have increases in frequency. So here, if you look at BMI, you see the insulin receptor substrate 1, the frequency of the polymorphism in this gene, which is denoted by this number, the RS, is the nomenclature for the actual polymorphism number. There's a catalog of this online that you can access the specific details of the sequence of this polymorphism. And lo and behold, the frequency of this polymorphism is very high on the people that had the good response where the BMI went down. So in that context, this is a good variant to have. It correlates with a steeper decrease in your BMI. But look at this other one, gamma aminobutyric or GABA receptor subunit alpha 2. that actually is not a good one to have as you go to the side of the BMI, which you don't want to be, which is where you actually gain or do not react to the exercise. There's actually higher frequency. Looking at HDL, you see for the HDL, we want to be on the right side of this. We want our HDL to increase. And here you have CC motif chemokine 2 that increases in frequency as you basically scroll through your cohort from the left side of the curve to the right side of the curve. And conversely, apolipoprotein B has the opposite behavior. So this is not one that you want to have for HDL because you actually have a higher frequency on the folks that resisted the increase on HDL. So that is the sort of yin and yang of physiogenomic analysis, and we can put this into a model. And in this model, it's a 40 by 40 model where we have the 40 markers that show the most consistent relationship to the phenotype. This is for body mass index. And then we take the 40 folks, 20 on the low side. These are the low responders. And then you have the high responders. So high responders would be the folks that actually lost the most weight. Low responders, those who did not gain weight or actually who actually gained weight. And then the markers that correlate with the positivity can give you a pattern. So everything that shows gray means that the person is heterozygous or has one copy of the gene marker. Black means that you have two copies or homozygous. And you can see that by the end of the day, this is a pattern recognition algorithm that can be calculated with specific coefficients for each member of the matrix. But it's a matrix made of 40 genes. And then predicting this can be applied by a combinatorial analysis of these different markers to see where do you fit in this profile. If you have an equal mix of these, you will be somewhere in the middle of the distribution. If you have an aggregation of markers on this quadrant, that means you're likely to be a high responder, and this will be a negative responder. But in terms of physiology, we're learning about the relationship among different pathways that perhaps may not have been obvious that were involved in this response, such as the chemokines. And so this gives an opportunity to learn from variability. So that can be then modeled where you say, okay, what is the contribution? If you're going to make this into a predictive model, how much can you contribute or how much can you predict from the baseline? And the baseline would be the phenotype at the beginning, before the intervention. And the answer is that 70% remains unpredicted, and the baseline can predict 30% of the variability between the HDL, the triglyceride, the cholesterol, and the fat. So you get 30%. When you add the genetic component, that gets you an additional 25. So you have 30 from the baseline, and you have 25% of the variability from the genetic component. So it is in terms of predictive power, the genetic markers give you a lot of advance. It doesn't predict the whole thing because we are not accounting for all the variability. There are other factors not accounted with regards to the baseline or the genetics, 45% to be exact in this model. But this gives you an idea of the power of the genetic analysis, but also of its limitations. It will never be the case that a genetic marker or a profile of baseline will give you 100% of the variability. But I think here you can see how they can support each other and make a more precise model by the end of the day when you consider these dimensions. So this paradigm has been very successful in the interventions that I described to you, and I am proposing this is a first conversation on this topic of precision medicine in regenerative biology, that this paradigm be extended to regenerative biology. And I will show you examples of how this has begun to be implemented in the field. I'm going to then go into implications of this for potential additional ways of understanding variability or reducing variability. And then lastly, I will bring some of the guidances of the FDA in regenerative biology to discuss how these precision medical approaches could impact the guidance that we have received in the industry so far from the FDA. So in the case of adipose stem cells, we can have the same issue of variability where you can consider the donor, the age, chronic diseases, basically what is the condition of the donor that provided those cells. And then we can measure aspects of the actual grafting in terms of where the sample was obtained, where and how is the sample obtained from the patient or the donor cultured and proliferated, and what happens to those cells when they are being cultured, the so-called secretome, in terms of what are the active elements of that culture. So we can begin by profiling each of these dimensions. In the case of a single donor, most of the profiling will be serum-based or serology-based. And here is an example of the kinds of profiling that we can do in regenerative biology. These pathways are complex. However, from an operational perspective, they are very tractable. I just told you the example with exercise was drawn from 222 genes from different pathways. So altogether, it's in the same scale as this map of immunoprofiling pathways. Many of these factors, many of these chemokines, interleukins, interferons, can be measured by ELISA, highly multiplex ELISA assays. So we can actually capture the phenotype of these pathways. Each of these genes, you can search for polymorphisms that are related to expression or to the actual function of the protein. So this diagram is perfectly suitable for a physiogenomic analysis similar to the one that I had utilized in the case of exercise and drug responses. This paper by Parsons et al., this is a group at Brown, I wanted to bring to this discussion because it is the closest I have seen to begin looking at the issue of donor. And here is a way this was done. So this group was looking at human adipose stromal vascular fraction, and they considered a series of patients. In this case, a small series, 13 patients to be exact, and they obtained half of the subjects were individuals that women that had recovered from breast cancer and were in remission. So the first 6 donors recovered from breast cancer. The other 7 donors had no cancer diagnosis. And they look at it from 2 perspectives. The first was the regenerative potential of these stromal fractions when they were cultured, and this is looking at 2 lineages. The first one is the adipogenic lineage, and the second is the chondrogenic lineage. And there were a series of measurements. In the case of the adipogenic, they were looking at intracellular lipids. In the case of the chondrogenic, they were looking at glycosaminoglycans. And this is showing before and after the response to the individual, and then the stars or the asterisks indicate that there was a significant difference. This group did not provide average or distribution plots. It's only 13 people, so it would not have been a particularly informative distribution curve. But nevertheless, if you let the eye do the comparison, you see obviously that there were many substantial differences among people with regards to their adipogenic potential or their chondrogenic potential. So, for example, you see that individual 7 had a very high adipogenic potential but very low chondrogenic potential. And then vice versa, you have individual 13 that had the opposite behavior. And so on. So you can start comparing, you can start doing relationships between these profiles, and there definitely was an observation that a major factor is the breast cancer status of the patients, but it was not enough. There was clearly inside each of these groups there was variability in the in either the chondrogenic potential or the adipogenic potential. They then look at the secretome, and they did a number of chemokines by ELISA. I'm showing you here 2. They did others, and here there was a stimulus. The cells were grown in minimum essential serum 3, and then the stimulus was tissue necrosis factor alpha and interferon gamma. So those were 2 different separate experiments. They were provided together to the quiescent cells to stimulate, and then the assay was done by multiplex ELISAs. And so the same thing, the 13 people, you look at the variability, and they had very substantial differences among each other inside each of the groups, the cancer versus the non-cancer. They also look at relationships. So some of the secretome elements were related, where you could see that a given interferon also correlated with a given chemokine, which shows that there may be some common sources of origin. So the data can be reduced in complexity by looking at relationships. Once you have multiple proteins secreted or multiple elements, you can do that kind of correlation analysis. So the thinking about this from a sort of design perspective, this paper, I think, set a very strong first pass or first attempt at characterizing this, and we could expand it by looking at the cells in the context of these different chemokines and growth factors. Pro and anti-inflammatory cytokines, anti-inflammatory growth factors, pro-angiogenic, etc. And all of these can be measured with ELISA, and you could have a layer of genetic analysis as well for each of these key genes. And the secretome, therefore, is a very good first step in approaching this. It's a reductionist approach where we actually would be profiling these different proteins that are being secreted. In this diagram, it's an approach at looking at the variability and trying to understand the variability. There is the component of secreted proteins. There's also the element of vesicles, of microvesicles that could be containing some of these active proteins, and this could be applied dynamically, where you look at different stages of the response to the adipocyte stem cell, and you look at different stages of disease, either the inflammation stage, proliferation stage, or remodeling. So there is a component of the pieces of the puzzle, and then the component of the dynamics of those as you move through different stages of therapy. At our institute, Dr. Lorenzin and colleagues have advanced the concept of engineering the secretome, and this is a very promising and very high throughput way of looking at the problem with modern technologies that allow us to undertake this. And it basically entails profiling of the secretome with a number of probes based on mass spectrometry, genetic sequencing, and Western blots, ELISAs, etc. So very high throughput, but very thorough look at the components of these universe of proteins and quantifying them statically in terms of who they are, which ones are these particles, but also importantly dynamically in terms of how they progress with time. This is a way of taking complexity, reducing it, quantifying it, and modeling it. It's very important that the potential of this initially clearly is for knowledge gathering, but there is a way of eventually moving this into therapeutics. Once we can reduce that complexity and understand it, you can even envision a situation where the components could be combined specifically into scaffolds that could be utilized for delivery. Different dynamics to the scaffold in terms of release. So the scaffolds could be collagen scaffolds, hydrogels, which are minimally invasive, or even microparticles, where the particles actually are loaded with the secretome. And this is our institute has advanced this idea of synthetic stem cells or self-mimicking particles that could be created once we know the information on the secretome. So understanding each of these elements of the secretome, I think it's a very powerful tool to understand the heterogeneity of these cultures. It's not getting yet into the donor issue, but it at least is addressing the baseline and the element of the phenotypes that are relevant to the specific responses. And the fact that it has a therapeutic potential as well, combining it with biomaterials, truly is a very powerful paradigm that deserves research and application in medicine. So I want to bring up to date some of these guidances with the FDA. I know that Army members have been exposed to this because Army is very strong on regulatory policy. I just wanted to review that again for this audience and then offer some perspectives on how these guidances interface with the element of variability that I was mentioning before. So we have been through this in our series. There is some controversy in our field. I think Dr. Rowe alluded to that in his talk, and it basically has to do with the two camps or the different ways of applications of regenerative biology, regenerative techniques in medicine, whether the application is at a clinic or whether the application is at an industrial level where the companies are developing these therapies versus clinical practices applying this to their patients. So this controversy in our field has been addressed by the FDA, trying to clarify it, and it has led to this initial guidance, which was in 2017, it is called the same surgical procedure exception. And what this was attempting to address was the fact that we utilize in medicine many procedures, surgical procedures that extract tissue and reimplant the tissue in patients. So coronary bypass graft, a vein is excised and then replanted in the cardiac vasculature to repair potential clots, potential improved flow, etc. But this kind of procedure is basically this same surgical procedure exception is saying this is not a FDA regulated process because it's the same donor. It's the same donor, it's the same facility, it's the same procedure. And so this would not be considered a regenerative product. Then in 2019, we had a breakthrough, and that is the RMAT, the Regenerative Medicine Therapies, Regenerative Medicine Advanced Therapy Guidance. And this provided a fast express line for biologics to be developed for the industry. And this has truly changed the map of the industry with this potential pathway to regulatory expediency. Once you submit your IND and you say that this is an RMAT, that will give you expedited access to the FDA, more guidance from the office, from the agency, and it will be, it really will be a sort of a badge of honor on the product that it has retained this this honor or this destination specification has a regenerative medicine advanced therapy. It applies to the devices, so the devices that go with a regenerative medicine therapy can also be put on the same rampway or expressway that from the actual product. It does exclude scaffolds. So scaffolds that are guiding the therapy will be part of the therapy, not considered a device. For example, collagen matrix that supports cells will be part of the product, not a device on their own. But for specific applications where there is a device that goes with the product, it would allow for an expressway for that as well. And this was February of 2019. And this brings us to this year, where in July of 2020, we got this final guidance. It was a revision from a previous one, regulatory considerations for human cells, tissues, and cellular and tissue-based products, minimal manipulation, and homologous use. So this is a very important summary and recopilation of the guidance and the strategy from the FDA. And I really think it's worth studying this, and I'll give you some remarks on details because it provides truly a regulatory path for regenerative medicine. It has this decision tree, which I think is fundamental in terms of how we can reconcile these tendencies in the industry. So let's go through this. So it's beginning to say, first, is your product the definition? Does it meet basically the definition of a regenerative product? Is it a regenerative medicine product of human cells, tissues, or cellular and tissue-based products? And it says if you can apply this surgical exemption, which is the same surgical procedure and location, it is not. And this is, again, coronary bypass graft, skin grafts, procedures that are done currently in medicine and do not imply any alteration of the tissue, but they do involve implantation in a different organ. But it's done in the same facility, same surgical procedure. So if it can be applied to the same surgical procedure and location exemption, it is not a regenerative product. Okay, so if it's not exempted, then we move to the next part of the decision tree. So the exemption, the same surgical exemption, does not apply. And here comes the minimal manipulation and homologous use considerations in this particular part of the Code of Federal Regulations. 27 CFR 1271, and you have 10A1 and 10A2. 10A1 with the minimal manipulation, 10A2 with the homologous use. And this is where the controversy has been, even approaching legal cases of the FDA going against some practitioners of the regenerative medicine. So if it does not meet the minimal manipulation and the homologous use, then you have to file an IND because this product is regulated as a drug device and a biological product, which means that an IND has to be submitted. If it does meet this minimal manipulation and homologous use, you do not have to file an IND, and the agency will use what they call enforcement discretion. Notice this guideline from 2020 extends that to the middle of next year. So it's not a free pass by any means. The agency may decide to enforce the regulatory or extend this discretion to further, but right now this is not exactly very far away. And so I think for these reasons, I really think that the growth of our industry has to be along this path, where we actually consider the product in multiple dimensions, in multiple uses. It may require different amounts of manipulation and introduction into organs different from where the graft was received from or obtained from, and that we should embrace a process that goes through a regulatory approval process. And that involves clinical trials, involves the use of profiling devices and techniques to reduce complexity, understand complexity, and model that complexity. And so here's a summary of these considerations. So the grafting or the material or the cells could be obtained from a variety of sources. We differentiate between the autologous and the allogeneic models. In the autologous, the recipient, the donor and the recipient are the same. In the allogeneic, it could be multiple donors to create a tissue bank and multiple recipients if it becomes an off-the-shelf product. Autologous so far had been done mostly at the clinic, and the allogeneic requires an industrial capacity to process and batch produce and file for the regulatory pathway to get the product tested in clinical populations and then submitted as an FDA product. This is a major driver for the allogeneic pathway, and that is the regenerative medicine advanced therapy incentive, what I call the expressway. To the extent that the RMAT can also include scaffolds as part of the product, it further enhances the speed of going into this rampway with the allogeneic model. Then you have to deal with the issues of the genetics of the recipient or the profiles of the self that gave from which the donor came. And so in this allogeneic route, it basically becomes a drug trial model in conventional pharmaceuticals. In fact, I was looking at some of the labels that have come out of this RMAT pathway, and sure enough, the sections on the label are the same as you see in drugs. Clinical pharmacology, side effects, special populations. The label is an FDA label, so it's the same format, the same categories applied to a cellular product. Now, there has been an interesting hybrid of these models, which I want to bring to your attention, because I really think this is a way of harmonizing these tendencies or these camps in regenerative biology that have been with some conflict and tension between them. And that is, there are models where a given biopsy, and this has been applied to chondrocytes. The case of chondrocytes is the one that probably is most advanced. The chondrocyte, the tissue is obtained by a surgeon, cartilage from the knee, and then the tissue is given to a facility, an expert facility, where the chondrocytes are grown, and then they are implanted in a scaffold, which is a collagen matrix, derived actually a porcine collagen matrix, and this product then is sent back to the surgical clinic where it is then implanted. So this process basically is a hybrid of a clinical procedure that begins with the surgeon extracting it from the patient, moving it to an industrial facility that provides the control, the quality control, the regulatory expertise, and the marketing and distribution expertise, and then sends back that to the clinic for insertion into the patient. It is considered a product, a regulated product, because it went through manipulation, expansion, packaging, depositing in a scaffold at the industrial site, but it landed at the same donor that gave the tissue, became the recipient. So in that hybrid model, you still will need very careful profiles of the immunity of the individual. There have been, as of October 2020, 47 of these RMAT designations. And I think there will be more, given the incentives provided by the agency. But I really want us in the industry to start thinking that the genetics of the recipient are going to play a very important role when you have products that came from multiple donors and now are being implanted in a specific recipient. This begins to look almost identical to a drug model where you have a medication and then you apply it to the first cohort of patients with a very clearly designed outcome that you measure. And so I think this is a major opportunity for us to start thinking of the genetics of the recipient, most of the trials that we're talking about are not the pharma type 10,000 people. It's going to be in that range that I mentioned before, the 100, 200, 300 individuals. And there you have the capacity of utilizing the physiogenomic approach that I described to understand the heterogeneity, learn from it, and predict it. So I want to thank you for your consideration. I mean, again, this was, I acknowledge this is a new perspective in the field, and this is more of a thinking piece. I want people to begin considering the ramifications of this. You can reach me at the email and telephone numbers here. I want to thank dearly the support of Dr. Cato Lorenzen, who is the director of our institute, and Dr. Nair, who is the associate director of our institute. They have provided a great home for me to develop these ideas about precision medicine in regenerative biology. My colleagues who were prior speakers in the series, Dr. Grady, Dr. Wu, Dr. Grimaldo, Caganello, and Dr. Rowe, again, are part of the expertise and resources that are available at UConn with our Army members. And then I want to acknowledge my previous collaborators at Harvard Hospital, Yale, and the University of Puerto Rico. These are folks that have worked with me for close to 15 years now, developing these different approaches ranging from drugs to surgeries to brain imaging, and hopefully we will be able to apply these also to regenerative processes in regenerative biology. And then support by grants at UConn. This work is supported by the Office of the VP of Research through the Convergence Awards for Research in Interdisciplinary Centers. And then I had prior support over the years from NIGMS, NIMH, and AHRQ. All of these agencies certainly have contributed to building this paradigm of physiogenomics and precision medicine. And then finally, since this is the last seminar in the series, I wanted to acknowledge the support of the UConn IT department. Kenny Blakesley and Blago Filipovich have been instrumental in making these presentations seamless to you. And then our marketing media manager, Melanie Burnett, in dissemination, and then at Army, Anna Stevenson and Terry Morelli, who have worked to disseminate these webinars through the community of members. Again, these webinars are recorded, so you can access them later at your leisure, and perhaps even extend it to other colleagues that may not have been able to attend the seminars. I would like to use this this time to consider some other factors that are important, and as we develop this strategy in the industry, one element of this is always a conversation with statistical analysis, statistical experts, as we incorporate biomarkers. The moment you introduce biomarkers, they have two purposes. First, they help you understand the biology, but they also can be utilized as covariates. And the moment you start talking about covariates, they could then be analyzed and be used to extract signal, extract responses, create subpopulations of effect. And that by itself is a major value of utilizing the physiogenomic approach. The second is with regards to implications in the marketing of the product or the distribution of the product. We had a situation in the early days of pharmacogenetics where there was a misunderstanding. Some members of the industry felt that the marker would actually reduce the market or reduce the range of patients that could get the population that could get the medication or the treatment. So it turns out that that was not the case. It never was the case. And what happened was that anything related to biomarker in terms of safety or efficacy was adopted by the FDA on the label. So, for example, the drug clopidogrel, Plavix, the FDA installed on the label a warning about the fact that since clopidogrel is a prodrug, you need an enzyme to activate it. In this case, cytochrome 2C19. So that is on the label of clopidogrel. But it's not as if clopidogrel had to be prescribed with a diagnostic. It was a warning, a guidance that was put forward by the FDA in terms of defining the population. And there are other examples where this becomes basically part of the surveillance, of the epidemiology surveillance that is part of the market surveillance that is a component of the regulatory oversight of therapeutic products. And it does not mean that the product can be administered only if you have this marker or the other marker. It becomes a guidance with regards to the follow-up. In the case of regenerative biology, we are not dealing with mega markets. I mean, these are going to be very defined biological conditions, pathological conditions, and so we are in a almost very similar to cancer, where you have micropopulations of high medical need, where you have to target the medications. That is very similar to what we face in regenerative biology. High medical need, very well-defined populations, and in that model, in that specific model, the precision medicine approach makes a lot of sense clinically, and also from a marketing perspective in terms of getting the products to the public. So I think the similarity of regenerative biology to oncology in particular makes it a very good specific field in which to apply precision medicine. So I look forward to any comments that you have, that you want to email me or call me, and we remain very active members of ARMY. So there will be an opportunity to interact through ARMY, through the different venues and the different events that the organization puts together. Thank you again for your attention. It has been a true privilege and pleasure to organize this series of webinars for the ARMY membership, and look forward to meeting you and interacting with you in new encounters. Thank you very much. This concludes the fourth seminar and the final in the series of clinical trials in regenerative medicine.